Hello everyone, my name is Vicky Ia, and I'm a graduate student in the bioengineering department, working under Dr. Living Tong. I'm going to be talking about the influence of lens power on IOL posterior lens capsule interactions and IOL's PCO potential. Now, we know that cataracts make the lens cloudy and obscure our vision. To treat that, they have to take out that cloudy portion of the lens and replace it with an IOL, or intraocular lens. That is a man-made lens which goes inside the eye to restore our vision. PCO, or posterior capsule opacification, is the most common complication following cataract surgery, and it results in clouded vision as well. So it's generally believed that lens epithelial cell growth and infiltration is what causes PCO. In addition, there's evidence that binding affinity between the intraocular lens and the capsular blag plays a major role. What this suggests is that with looser binding IOLs, there's more space between the IOL and lens capsule, okay? So cells will be able to infiltrate more easily and then proliferate and differentiate, which causes PCO. Tight binding, on the other hand, would cause low space and prevent cells from infiltrating that space and result in less PCO. So what we're most interested in is how do these phenomena affect patients with myopia? Now, myopia is a growing problem. It's expected to double by 2050, and unfortunately, it's been found that PCO can be a particular problem for severely myopic patients. These patients use lower power IOLs to correct their vision and have been observed to have a 30% clinical PCO incidence. Meanwhile, lower myopia patients that use the mid and high power lenses have been observed to have a lower PCO rate. It's also been observed that IOLs for myopia patients have weaker interactions or adhesion towards the lens capsule. Now we must ask ourselves, are these two phenomena related? Severely myopic eyes, again, they only need low power lenses to correct their vision. And interestingly, in terms of their geometry, these lenses are flatter, and the high power lenses are more convex. So we can quantify this geometric feature using posterior radius of curvature. This concept, called PRC for short, basically when we look at the plot of our PRC versus our powers, with lower power lenses, you actually have a higher PRC, and with the lower power or higher power lenses, there's a low PRC value. So in other words, if you were to trace a circle around the bottom curve of that low power lens, for example, you would actually get a circle with a high radius, and conversely, with the high power lenses that are more convex, you would have a radius that's much smaller, right? A low PRC value. Okay. So knowing that the myopic eyes are treated with these low power IOLs with high PRC values, the question we ask is, does IOL power or its associated geometry have a significant impact on its affinity for the PLC and therefore impact PCO potential? How do we test this? To test it, we chose a 3D in vitro model with the original shape and dimensions of the adult human lens. The way we made this is by first designing this 3D print and using that to cast the shape of the lens into molten gelatin. Okay, and this results in what we call a simulated posterior lens capsule, sPLC. And then with that, we can place the IOL inside and do tests to understand how the IOL and PLC interact physically. It also allows us to study how the cells respond. So in terms of the IOLs chosen for the study, we did 3 IOLs that are all commercially available in 3 different dioptric powers. So we have 6, 20, and 30 diopter IOLs. And these represent the high, medium, and low myopia, respectively. The 6 diopter IOL has a high radius of curvature, and because it's the flattest, the 30 diopter one is the lowest PRC because it's the most convex, right? With this model, we then hypothesized that the lowest power IOL, 6 diopter, would result in lower binding affinity, increased space, and higher cell responses than the 20 and 30 diopter IOLs. On the other hand, 20 and 30 diopter IOLs would have high binding affinity, minimal space, and therefore minimal cell infiltration. Is this true? So to answer this question, we studied the interactions. How did we study the interactions? We studied the space and the percentage of contact between the IOL and PLC, okay? So we used a method called OCT, optical coherence tomography, and this uses light interference to discern between the different materials and tissue types, right? And gives us this cross-sectional image. Specifically, you can see that the 6 diopter has a huge amount of space, while 30 diopter has a lot less. When we quantify that space, 6 diopters had much lower area underneath them over time, and there's not much difference between the 20 and 30 diopter IOLs. IOLs with large space also led to a small percentage of contact. So is the physical contact the result of the binding affinity between the IOL and the sPLC? To study the binding affinity, we used a custom microforce testing apparatus to measure adhesion force between the IOL and sPLC. A bending bar is attached to the IOL, and this motorized stage moves downward, causing the bending bar to deflect, and a camera is there to capture the distance that the IOL had traveled before it detaches from the sPLC. Okay, and then we calculate the adhesion force using Young's modulus of that bending bar and the formula you see here. What we found is that 6 diopter lenses had the lowest adhesion force compared to 20 and 30 diopter, and there's no significant difference between 20 and 30 diopter IOLs. The next question is, will the large interface space allow more cell infiltration? To test this, we used lens epithelial cells and seated at the edge of the IOL optic. Through the OCT imaging, we were able to observe that for the IOLs with more space, the 6 diopters, LECs were able to infiltrate more than with the 20 and 30 diopter, which were impeded from infiltrating. We then quantified the cells along different points along the edge, from the edge to the center of the IOL, and saw that there was significantly higher cell density for the 6 diopter IOLs than for the 20 and 30 diopter IOLs. As expected, large space was associated with strong cell infiltration. So does space have a similar effect on cell proliferation and differentiation? The answer is yes. In terms of the 6 diopter with large space, we also saw stronger cell proliferation and more differentiation. To conclude, it has been observed that high adhesion force between IOL and PLC leads to lower interface space, which leads to lower levels of cell response, and reduced PCO potential. However, in the case of severe myopia, it is totally the opposite. Their low-power, flatter IOLs led to lower adhesion force, more space, which allowed high levels of LAC infiltration, and that is why they have a high chance of PCO. With this information, it's possible that we can modify the IOL design to improve binding affinity and reduce PCO potential and improve IOL safety. Thank you very much for your attention and the opportunity to present my work.